Well, good morning, good afternoon, everyone. I'm Paul Danis with the consortium, and pleased to have you join us for another edition of Proteoform Thursday here. Today, we're happy to have Melissa Bärenfänger with us, who's going to enlighten us on glycoproteomics. So let me just give a few brief news items, and we'll get right to her talk in a second. So first off, we'd like to thank all of our sponsors enabling us to do these kinds of programs and grow the whole field of proteoform biology. We're very thankful to our partner SEER, as well as our major sponsors, Thermo Fisher, Bruker, SciEx, and Lilly, as well as our supporters, Agilent, Pfizer, and Nuomics. And if you or someone you know is interested in supporting the consortium, we'd be happy to hear from them, and they can contact me or any of our colleagues on the board. So one thing that some of you may know, we're excited about our symposium we're going to have this October in Chicago. The registrations are now open, and we have a lot of the speakers up there, and abstract submissions will be opening up next month, and we'll have a specific announcement going out to all of you on that shortly. So keep your eyes tuned on there. We have a number of really exciting speakers. Some of you probably know a lot of these folks here, and we're really going to cover a lot of the topics dealing with, top-down methodologies, top-down use in clinical applications, as well as some of the newer technologies coming out as far as the non-MS approaches to doing proteoforms. So it should be pretty exciting to see kind of where the field's evolving and how we can look at these intact proteoforms and really understand the biology that's associated with them. Additionally, if you or some of your colleagues are interested in upping your knowledge on proteomics through top-down approaches, at ASMS, beginning of June, there's 2 courses. It's a Saturday course dealing a lot of the methodology of how to do it, and the Sunday course is specifically around data analysis. They're 2 separate courses. You can take one or both. But one thing to note is that the advanced registration deadline is a couple days. So if you're a colleague or interested, please go ahead and sign up for that as soon as you can. That should be quite useful. They did it last year, and they had really, really good interest in the course. So that's moving forward. So please spread the word on that if you haven't already. So also, please join the community, sign up at the website so you can be first to know about all of our news and activities going on in our initiatives. You can follow us on Twitter at Top-Down Proteome and LinkedIn as well. We have some sites there. So next, Byron Drown from Purdue will be speaking next month at the end of May. So we'll have details on that going out to you as well for registration and timing, etc. So now let me turn it over to Dr. Melissa Bärenfänger, who's with the Amsterdam Institute for Molecular and Life Sciences at VU Amsterdam. And she's going to speak about pushing high mobility MS to understand glycoproteins, from increasing glycoproteomics IDs to observing glycoprotein unfolding. So go ahead, Melissa, it's all yours. Thank you.
Hi, thanks, Paul. I will share my screen. And would like to say thank you again. Thanks for inviting me to speak here today. I would like to talk about glycoproteins today, and I like glycoproteins a lot. I've been working on glycoproteins for quite a while right now, and how we can use mass spectrometry, in particular eye mobility, to study these complex proteins. But before I start, I just want to mention that here in the Netherlands, today actually is King's Day. And King's Day is a day where we celebrate the birthday of our king. So I'm happy that I see that a few of my Dutch colleagues are here at this talk, and nonetheless. And I also would like to use the opportunity today to highlight not only my research, but also a few other groups working in the Netherlands and doing some great research on proteins, and particularly glycoproteins. So before I start, I just want to briefly tell you who I am, why I'm here. I did my PhD actually in Germany, so I'm German, I'm not Dutch, but still I'm celebrating King's Day. And in that time, I was working more on analyzing intact proteins, intact glycoproteins, and to see how these glycosylation of these proteins can change and how we can detect them with mass spectrometry. After that, I came to the Netherlands and did my postdoc in Nijmegen at the border to Germany, where we developed glycoproteomics methods to study changes in a lot of glycoproteins in a clinical setting. And the work that I'm going to present today is also partly from that time. And since 2022, I moved to Amsterdam, and I'm now assistant professor at the Vrije Universiteit in Amsterdam at the Division for Bioanalytical Chemistry. And here I want to focus on also, of course, glycoproteins and how we can understand the structure, function, and relationship between changes in glycosylation and glycoprotein function. All right, so my talk today will be a little bit a mix of some research I've been doing during my postdoc and some things I want to establish here in Amsterdam. So I have a slide on today's content just to give you an idea what this is going to be about. So since this is a top-down proteomics consortium, I first want to say I'm not going to present something on top-down today. I will present some data on bottom-up proteomics and how we can use, in particularly the timsTOF Pro to analyze glycopeptides. And I will also tell something about an application of this method. So this is more on bottom-up, so on smaller peptides and glycopeptides. But then I would also tell something about how we can study intact proteins with mass spectrometry and how we can start to unravel also structural aspects or functional aspects of glycosylation on proteins. So I don't have a long introduction because I think you're all aware that proteins are fascinating, and also the concept of proteoforms is very important. And I just want to give you a brief overview on what actually glycoproteins are. So glycoproteins are proteins where we attach carbohydrate structures. These carbohydrate structures can be very complex structures and can look something like this. The reason I'm interested in glycoproteins is really that if we attach these complex carbohydrate structures to proteins, we really create a huge number of different proteoforms. So there's really a vast heterogeneity going on. And I think that glycosylation is really a challenging PTM to look at because, yeah, we can have so many different branches, we can have so many different structures attached to this protein that really it gets very fast, very complex. And in my presentation today, I want to focus more on N-glycosylation of proteins. And this is the attachment of carbohydrate units to an asparagine side chain. And what I will do is use this nomenclature that you can see here. So even though carbohydrates are very complex, have a lot of functional groups, usually we try to make our life a little bit more easy and try to, yeah, just describe them somewhat like this. So from these structures, we often have the compositional information, what kind of monosaccharide units are linked to our protein. But if we would look closer, of course, beneath these structures, there are even more isomers or more proteoforms that we can have. So why glycoproteins? In which areas are they important? And I brought a few examples. The attachment of these sugar units is really important for the signaling, for example, cell-cell signaling, or glycoprotein interactions. We also see that these changes in the glycosylation of proteins happens in all kinds of diseases, meaning that we can also use them as a biomarker for the early detection of various diseases, for example, cancer disease, Alzheimer's disease, and so on. Glycosylation also plays a very important role in our immune system. We know that if we have antibodies, for example, and they have a specific glycosylation, the immune response can be regulated by that. So a lot of people not familiar with this world often see this glycosylation as making the protein more soluble, and that's kind of it. But for me, that's really not the case. So for me also, I see the functional aspect of these glycan structures, and we have a lot of receptors, we have a lot of lectins that really specifically recognize distinct epitopes. So the attachment of these carbohydrates to sugars is not really only for solubility, but really creates proteoforms that have different function. So there are different ways to study glycoproteins, and the first challenge, of course, before understanding what is going on in this broad context is to define the structure of a glycoprotein. And I don't have to tell you that much. I guess you're aware that, of course, you can study proteins or glycoproteins as with intact mass spectrometry or top-down mass spectrometry. You can go for a middle-up approach, you can go for a bottom-up approach, or you can also go for release glycans. And again, today I will first go into the bottom-up approach, and then later on I will also talk something on intact protein MS. So I would say if we're looking at proteins, then the field of proteomics is rather big, and there are a lot of instruments, a lot of methods developed to do a proteomic screening. However, if we want to look more into changes in glycosylation, then our life becomes a little bit more complex, and we might not be able to use the same bottom-up techniques that we use for, I would say, regular proteomics approaches, but we need to consider a few things. So I want to start with the first chapter of my presentation and to tell you more on what we need to consider when we want to make glycoproteomics work, in particular in comparison to proteomics. And how can we use ion mobility to get even further with our glycoproteomics approaches? So proteomics is well established, but if we want to focus more on glycopeptides, we have to consider that glycopeptides are usually bigger than regular peptides, right? Because they have this huge glycan part attached to them. We also need to consider that glycans, unfortunately, if we analyze them with mass spectrometry, tend to break down more easily than the peptide backbone. So that can make our life a little bit more challenging because our MS settings might break them down before we can detect them. And also glycans don't really like to ionize in positive ion mode that much. Usually a lot of the proteomic settings and also in combination with chromatography are tailored towards positive ion mode. Also, if you combine this, for example, with reverse phase chromatography. And if we go for the standard approach, then we really need to consider that ionization efficiency might not be as great for these glycopeptides compared to regular peptides. And there's another challenge, which is that if we use CID fragmentation, so collision-induced fragmentation, then usually what happens is that the glycan part breaks down very easily because it's just much more fragile, but the peptide part stays mostly intact. And the problem with that is that if we don't know the peptide sequence, we also cannot tell really where our glycan part is coming from. So from which protein it is coming from, or what is the position in a particular protein. So what we really need to consider is how can we have information on the glycan part attached to a peptide, but also on the peptide backbone. And with CID, sometimes that is a little bit more challenging. So I'm not the only one trying to figure out how can we make this happen. And I mentioned that I want to highlight some research from the Netherlands because today is King's Day. And of course, I want to mention the group in Leiden at the Leiden University Medical Center. And they already looked into this problem that glycans don't really like to ionize in positive ion mode that much. To help this a little bit, they use during electrospray ionization dopant-enriched nitrogen gas. Here in the study that I have on the left of the slide, they are looking at release glycans and in negative ion mode, but also in negative ion mode, the dopant-enriched nitrogen gas can help to increase ionization efficiency of glycans. Not from the Netherlands, but also a very nice study is from Daniel Kolarich and his group, and he actually was looking at glycopeptides and saw that if we enrich our nitrogen gas during electrospray ionization with acetonitrile, then we increase the ionization efficiency of glycopeptides quite a lot. That can also work for peptides, but it even works better for glycopeptides. So this is one way of tackling one of the issues we have when we want to study glycopeptides. But of course, there were more challenges, and another group that I want to mention here is also from the Netherlands, from the Utrecht University, and they recently had a very nice paper on studying glycopeptides with the timsTOF Pro, and they tackled two challenges that I was mentioning. The first one is that glycopeptides are bigger than regular peptides, and we can actually use this to our advantage, because if glycopeptides are bigger than regular peptides, it is much more easy to separate them from regular peptides during high mobility mass spectrometry. And the second thing they tackled is also that the collision energies for glycopeptides needs to be adapted in comparison to regular peptides. So by increasing collision energies, you can also have fragmentation of your peptide backbone to really also get information on the whole glycopeptide. So there are already a few studies that show you tips and tricks in what to do when you're setting up your glycoproteomics experiment. And what we're trying to do is basically combine all this information that is out there into an ultimate method to study glycopeptides or to identify glycopeptides as best as we can. And I have this overview slide of our setup, which is quite simplified. So for this study, we also use the timsTOF Pro coupled to a nano LC system where we first separate glycopeptides and peptides, and then use a nanospray source, the captive spray source, prior to electrospray, prior to infusing our analytes into the timsTOF. We were looking into also reducing gradient times, but I will not talk about this today. And then we tried to combine different aspects of improving glycopeptide identification. So for example, we also looked again into this dopant-enriched nitrogen gas to see how much more identifications we get. Since we have been working with the timsTOF Pro, we will also look into the high mobility aspect, and I will come to this later. And we also were working on optimizing the collision energies. So what we did is basically we started with a test sample, with a very complex test sample. In this case, we used human plasma, and then just did a triplet digest to obtain peptides and glycopeptides from this very complex sample. We did a glycopeptide enrichment to really have high abundance of glycopeptides in our sample. Then we did our LC MS experiment on the timsTOF Pro with a nano LC, recorded MS data, and for data interpretation we used MS-Trager Glyco. MS-Trager Glyco works in a way that you can submit your data files, and you can also submit a list of glycan mass offsets. And then the algorithm will, based on the glycan mass offsets and the fragmentation data, match glycopeptide identification to your data, to the data you submit. And what we really wanted to see is which parameters do we have to tune to increase the identification of our glycopeptides. Also in our sample, even though we did a glycopeptide enrichment, we still had some peptides left. So we also wanted to see a little bit, okay, if we increase the glycopeptide identification, how are our peptides doing? Is the identification of the peptides going down, or is it also better for the peptides? But actually, which parameters do we need to tune to identify as much glycopeptides as possible? Why do we want to do this? Of course, if you think that, if you can think that biological samples such as human plasma contain huge number of proteins, huge number of glycopeptides. So we really want to dig deep, really want to identify as much as possible to characterize the whole the whole glycoproteome in these complex biological samples. So first, I want to talk about, I basically want to go back from the back to the instrument to the front. I want to talk about the collision energies that you need to adapt when moving from proteomics to glycoproteomics. And in the timsTOF, you have the you can ramp your collision energies not based only on the mass to charge values of your ions, but also on the mobility value of your ions. And what you can see here in the heat map is the distribution of your ions over the mass to charge dimension that you usually have, but also in the mobility dimension that you have on iMobility instruments. So I would say standard proteomics methods usually have collision energies from 30 to maybe 60 electron volt to fragment peptides and to get a nice peptide sequence coverage. However, this does not work well for glycopeptides. So I brought you an example of one glycopeptide that you can see here on the right. If you measure this glycopeptide with the standard proteomics settings of this instrument, then you get fragmentation data that looks like this. So that doesn't look great, right? You don't have a lot of fragment ions. You will never be able to really identify the peptide backbone or the glycan part of this glycopeptide. So for this precursor, which is a 2+ precursor in the proteomics standard settings, you get a collision energy of roughly 60 electron volt, which is, I would say, already on the high side, but still not enough to fragment down glycopeptides. So of course, we increased the collision energies and came to eventually ideal settings. And in these ideal settings, we use collision energies of roughly 19 electron volt for this 2+ precursor. And you can see that we get a nice fragmentation of the glycan part, which you can see here on the right side, where you see peptide and glycan fragments. But we also detect some oxonium ions, which are just breakdown products from the glycan. The same glycopeptide also ionized as 4+ precursor, and here you use less collision energies to get the same effect. Of course, the higher the charge state of this glycopeptide, the more likely it is to break down. So we can use here less collision energies to achieve the same effect. So there's one thing I want to mention here, because you see that, for example, for the 2+ precursor, we already use quite a high collision energy of 19 electron volt. In the timsTOF Pro, your collision energies are, your maximum collision energies that you can set are 100 electron volt, unless you talk to Bruker and ask them to increase these values. What we did, so in our method, we can also choose to go higher than 100 electron volt. And I have two figures more on this here. On the left, you see how we ramp the collision energies up as a percentage to the standard proteomic settings, and how that really increases the number of our glycopeptide IDs. At the same time, if we really go high with these collision energies, you also see that the number of peptide IDs slowly goes down, but we are still able to identify also some non-glycosylated peptides. On the right, you see a figure where this is again shown, but then for individual charge states of these glycopeptides. And also, as expected, this effect is more drastic for charge states that are lower. For example, the 5+ charge state barely shows any improvement. However, especially for 2+, 3+, 4+, we see that the collision energies really help to get more identifications out of our data. So the next thing I want to talk about is the TIMS funnel and what you need to consider here when you want to identify glycopeptides. And in the timsTOF Pro, you select your ions for fragmentation not only based on mass over charge, but also based on the mobility dimension you have. So your precursor selection really is a square based on a specific mass over charge width and a specific mobility width. And this is also something that we need to consider when comparing regular peptides to glycopeptides, because I already mentioned that glycopeptides are, of course, usually larger than regular peptides because of the glycan moiety that is attached. But what we also see is that glycopeptides just have a broader mobility than regular peptides. You can imagine that this glycan part is very flexible and can have a lot of different conformations. So we also see that in mobility. And for these glycopeptides, we usually observe a way broader mobility distribution than we observe for the peptides. So this is also something we looked into. Basically, what is our optimal window or optimal mobility width for this precursor selection? And you can see here in this figure that if you increase your mobility width for precursor selection, then that you also really increase your identification of glycopeptides up to a certain point. If you're too large, eventually this will drop down again. And well, for the peptides, we just lose peptide identification. The standard proteomic settings are here, the first data point that we have. So roughly 0.02, 1 divided by K0. And we really broadened this quite a lot to have more glycopeptide IDs. The idea behind this is that if we have a broader mobility window, we will have more ions to fragment. And of course, if we have more ions to fragment, then we have a better quality of our MS2 data. So basically, by choosing a too narrow window, we lose half of our ions that are not fragmented. And the quality of our MS2 data is not as well as having a proper mobility window. All right, the last aspect is the dopant-enriched nitrogen gas that we use. And I only have one slide on this to show you basically what we gain with all the improvements that we made. And on this last slide, I just want you to show how we really optimized these settings and what was the gain that we have. So on the two top figures are the standard proteomics settings that you get when you buy the instrument. On the left, we didn't use any dopant-enriched nitrogen gas, but just air. And on the right, we used acetonitrile as enrichment. And in this figure, you can see the number of identified glycopeptides in their master charge and mobility dimension. And you see that if you use just the proteomic settings and also no enrichment of the nitrogen gas, that you identify barely a handful of glycopeptides. This is already helped a lot if you use dopant-enriched nitrogen gas. So then already you increase the number of identifications quite a lot. I think mainly because the ionization efficiency increases, but also because you increase or you push your glycopeptides into higher charge states and you get better quality MS2 data. However, if we consider all the parameters that we optimize, you can already see that even if you don't use dopant-enriched nitrogen gas, you identify quite more glycopeptides compared to the standard proteomic settings. However, if we combine everything, our optimized settings and the use of the dopant-enriched nitrogen gas, the number of glycopeptide identification is really tremendously increased. Okay, so now we have our method set up. And of course, we want to apply our method to a complex problem. And I use this method to study glycoproteins from CSF of patients diagnosed with congenital disorders of glycosylation. So this was a complex challenge because, well, there are again problems. If we're working with CSF or cerebrospinal fluids, then the first thing that is a challenge is that in this sample material, the protein amount is very low. So you need to be very sensitive. Second challenge is that this CSF sample, also similar to blood or plasma, contains mostly albumin, a protein that is not glycosylated, so a protein that we are not that much interested in. And the third challenge is that some proteins in CSF originate from the brain, and some proteins from CSF originate from the blood entering the CSF via the blood-CSF barrier. So you also get a mixture of proteins that are partly representing the glycosylation status of the brain and that are partly representing the glycosylation status mostly of your liver. Another interesting fact is that actually the brain makes a different glycosylation than our liver. So there is something called brain type glycosylation, showing again that this process of glycosylation is very tissue dependent. And the glycans we see in proteins that are purely derived from in the brain are structurally different from your typical plasma type glycosylation. All right, so why were we looking at congenital disorders of glycosylation? It's a disease that involves genetic defects somewhere in our glycosylation machinery. So you have a very complex figure here on the left, depicting protein glycosylation and protein biosynthesis in the ER and in the Golgi. There are a lot of enzymes involved in glycosylating a protein, enzymes that are involved in creating the precursor monosaccharides, enzymes that are involved in transferring oligosaccharides to the newly formed protein, and also a lot of enzymes that trim the protein and attach different monosaccharide units. So whenever you have a genetic defect in any of these processes, it is considered a congenital disorder of glycosylation. And what happens is, let's say we have a genetic defect here, this protein doesn't do, this enzyme doesn't do its job anymore. Usually it has very, very severe consequences for the patient. And what we see quite often in a lot of these patients is that they have neurological abnormalities. So their brain is underdeveloped and they really suffer in various ways, but usually some kind of neurological dysfunction is involved. So we were wondering, of course, it's a genetic defect, so every organ should be affected, but why is the brain specifically so much affected from this compared to maybe other organs that are involved or that also have this genetic defect? So that's why we were looking into the CSF samples from these patients to figure out if maybe the glycosylation defects we see on brain-derived protein are different or more severe than the glycosylation defects we see on in plasma or on blood-derived proteins. So sample prep is a little bit similar than compared to the sample prep we did for our method development. In this case, of course, we take CSF samples and not plasma samples. I did a quick protein enrichment just by precipitation, tryptic digest. And then in the previous method development, I told you that we were doing a glycopeptide enrichment. However, in this case, we did not perform a glycopeptide enrichment for two reasons. Of course, one, we have a very good method that identifies glycopeptides very well. But the other reason is that specifically in these patients, we expect to see highly truncated glycan structures or even absent glycan structures due to their genetic defect, because they are not able anymore to really glycosylate the proteins that they have. So if we would enrich our glycopeptides, we might lose these highly truncated forms, and we can also not detect anymore if the glycosylation is completely absent. So that's why we didn't perform any enrichment here. So again, the measurements on the timsTOF Pro with the nano LC, and the data interpretation was also done with MS-Traeger. And before I show you the results of the patients compared to control, I want to show you this figure here, which is only the distribution of the glycoproteins and their glycosylation in healthy individuals. So we detected a lot of proteins, a lot of glycoproteins in these samples. And based on genetic information, we can roughly estimate which of these proteins are mostly coming from the liver and entering the CSF via the blood-CSF barrier, and which proteins are mostly produced by the brain, and therefore reflect the glycosylation status of the brain more accurately. And you can see here that what I mentioned earlier is that the glycosylation profile in plasma proteins is very different than the glycosylation profile of brain-derived proteins. And also here on the side, I just want to mention another, again, the group in Leiden, which did a very nice review paper in 2016, where they looked into all publication on plasma proteins and came up with roughly the same profile that we find here. So that was a nice confirmation that our method works, and also that we create enough data to reflect the overall glycosylation status. However, of course, we also always have the information on which protein, from which protein is it coming from. So in our CDG patients that have really issues with the glycosylation machinery, we see also this on our glycopeptides. So in this figure, you see groups of glycans. So for example, the complete loss of glycans or highly truncated glycans, either on blood-derived proteins or on the brain-derived proteins. And of course, we had a lot of different patients with different genetic defects that are maybe not always comparable. So I want to highlight just two of them, two of our patients that were most severe, so the ALD6 and the PMM2. And for these patients, we see that they lose their glycosylation on a lot of proteins. However, this seems to be more severe on the brain-derived proteins than on the blood-derived proteins. So here for the brain-derived proteins, for 15 to 20% of the proteins, they completely lose their glycosylation, whereas on blood-derived proteins, this is only maybe 7 or 8%. So of course, yeah, we to really say something about this, we need a larger sample size. However, it was the first indication that there might be a difference. And it kind of makes sense considering that even though it's a genetic defect, glycosylation is tissue dependent. So we will see also a slight different effect on how this genetic defect plays out, either on brain-derived proteins or blood-derived proteins. And I think a lot of studies basically stop here. And it's important work that we need to do. So we really need to define what kind of structures are changing, why are they changing, how are they changing. But I also want to move on now and really ask the next question, because now that we have detected these differences, what does it mean for the protein? And in the end, what also does it mean in the bigger picture? So of course, if we're talking about protein function and how glycosylation affects protein function, we cannot do this on glycopeptide level anymore, because we break down our proteins, and of course, then we cannot study the function anymore. So I want to move back to studying intact proteins, and in particular, how we can use also immobility here to study glycoprotein stability. So what we saw in our patient samples is that in the severe cases, they lose their glycosylation. So what we were trying to do is look at individual proteins, try to recreate this phenotype enzymatically, where we cut off glycans and see if that would affect protein stability. So we were looking at one protein in particular, which is transferrin. Transferrin is a rather large glycoprotein of 80 kilodalton and has 2 glycosylation sites. And if we look at transferrin from healthy individuals from CSF, we see that, and we measure this as intact protein, we see that we have one dominant peak corresponding to a proteoform where we have two glycans attached. And this is basically what we see mostly in healthy individuals. If we look at our patient that has a genetic defect in the glycosylation machinery, we see that the patient still has some proteoforms corresponding to the healthy phenotype. However, we also see the loss of one glycan or even the loss of both glycans on the protein. So to study this in more detail, we were creating this phenotype enzymatically, or recreating this phenotype enzymatically by using PNGSF. And you can see that we were quite successful in cleaving off one of the glycans and also a little bit cleaving off both of these glycans. So if you want to study stability, of course, you need to do this in a native way. So if you ionize transferrin with native electrospray mass spectrometry, you get a spectrum that somewhat looks like this. In our sample, we have the native proteoform with two glycans attached, and also enzymatically recreated the proteoform where we lose one glycan. So you see that for both of these proteoforms, we observe charge states from +19 to +21. And now what we can do is actually with eye mobility mass spectrometry is to mass select these glycoproteoforms and do collision-induced unfolding experiments on these glycoproteoforms separately. So we have in our lab in Amsterdam two instruments to do this. And I quickly just want to mention the photosynap that we have, which is capable of performing these studies. And we have an eye mobility cell here. And what we can do is actually induce stress on our protein, for example, in a collision cell. And the more stress we put on our protein, the more it will unfold, depending on how much we stress it. If it unfolds, we will, of course, be able to detect this unfolding in the eye mobility dimension, because since the shape will change, also the mobility of this protein will change. And I also want to quickly mention that actually in our lab we have the photosynap, so we modified it to also do IR and UV spectroscopy on molecules, which is in collaboration with MS Vision. So we have the photosynap with the TWIMS eye mobility. We also have the timsTOF with TIMS mobility. And here you can do similar experiments where you don't stress your proteins in a collision cell, but actually you can stress your protein in a TIMS funnel and start to see how it unfolds in the TIMS funnel. And if you compare this unfolding between these two instruments, you see that in the TIMS funnel, this unfolding doesn't happen as well as in the real collision cell. But to talk about this a little bit in more detail, so what we did is we see the different charge states of the transferrin. And in case of the TIMS, we increase the potential, we stress the protein by increasing the delta potential in the TIMS funnel. And when we're looking at different charge states, we see that, okay, in lower charge states, we cannot really stress this protein, and the protein has a very constant eye mobility. But however, in higher charge states, we slowly see this unfolding happening if we increase the stress in the TIMS funnel. In the TWIMS, where we use a collision cell, we are also able to unfold the lower charge states. I have here a figure on the generated on the TIMS, and the idea was that we want to look at these two glycoproteoforms separately. So we want to really figure out if we stress the protein with only one glycosylation site, does it unfold differently than the protein that has two glycosylation sites? So we did these collision unfolding experiments for both of these proteoforms, and you can see their plots here. So by increasing the potentials in the TIMS funnel, eventually the protein starts to unfold for both glycoproteoforms, so with one glycan, but also the one with the two glycan. However, their unfolding is slightly different. And in the difference plot, you can see that specifically at higher stress levels, at higher potentials, that the different glycoproteoforms unfold differently. And I think for me, this is the first indication that glycosylation actually plays a role in proteoform stability. I think this is a first step in also figuring out if small changes in glycosylation change the stability of a protein, how does it change the function of a protein? So that is actually what I want to do more in the future. I want to develop mass spectrometry methods to study proteoform-specific structure and function. I think also that we need to have methods that really link these structural changes to functional changes. So maybe also combining these mass spectrometry experiments with functional assays that are ideally proteoform specific. And I showed you now work on congenital disorders of glycosylation. In the future, I want to move more towards neurological diseases, in particular because I'm interested in also figuring out why we see this specific brain type glycosylation that is so much different from the serum type glycosylation. So lastly, I want to acknowledge a lot of people that were involved. I want to acknowledge my old lab in Nijmegen and the Repro Consortium for Glycoscience, but also my new lab here in Amsterdam. And yes, I'm open for discussion.
Great, thank you very much, Melissa, for the tour of the world of glycoproteomics. There's definitely some challenges there. I had a question. If folks have questions, they can either ask it in the ask it live if you want to raise your hand, or you could send something in the chat and we'll be happy to deal with it. I just had a quick question. When you talk about the unfolding, do you have a sense as to what physically is happening as this thing unfolds? Is it like one giant piece flaps over, or it's a whole thing just sort of getting bigger? Do you have any sense physically what's happening during the unfolding process?
So I think it is, of course, induced by some kind of collisions. So I would think that we somehow break down the three-dimensional structure, or that the structure slowly starts to unfold. And looking at the mobility, I think these structural changes need to be big. Since the mobility of the whole protein is already so broad and we see a huge shift in mobility, I think really that globally something is happening.
So they're pretty significant changes, not like just a couple
I don't think that
flopping over.
I think if it would be only a small change, then we probably wouldn't see a huge difference in mobility on these big structures. So I think something, a real unfolding event is happening.
Great, thanks. Moe, you have a question?
Yeah, very nice work, Melissa. It's very nice to see the data on the team stuff. I have like a general question. I really like your last comment about trying to connect with some kind of physiological assays. And you mentioned like if we can have assay that's proteoform specific. So that's also like my question is like I know like mass spec is really powerful to really resolve all the different states, but I don't know if there's anything that's kind of you can easily find to be able to connect like to like a proteoform specific assay to kind of have the downstream biological effect.
Yeah, I think as you mentioned, mass spectrometry is very powerful because we can at least be mass selective, right? So we can at least select proteoforms en masse. And then I think, well, we have to do it the hard way and really try to isolate proteoforms. for example, in a chromatographic way, or affinity-based, or however you think would be the best approach for the proteoform of interest. It is a challenge.
Yeah, it's like it's hard for me to envision something that's kind of like exactly parallel with what mass spec measures. So it's kind of like a little for the functional assay. So I also have like a related, more like a philosophical question. I was wondering, like in nature, I guess maybe there is always some kind of distribution of glycans. Like I don't know, like in biology, like how precise that glycan has to be for like a specific biological function, right? Is there always just some kind of redundancy of like some kind of like a heterogeneity always, just to have to be like a flexible in some kind of binding or something? Do you have any thoughts on that?
I think that's a very good question, and I'm not sure if I can give you the perfect answer to that. I know that some processes are probably not the most specific ones. So for example, what we see in humans is that usually all the glycan structures are carry sialic acid at the end. If a protein gets old, it starts to lose the sialic acid, and then the galactose gets recognized by certain receptors and tell our body that this protein needs to be degraded. So, for example, this process is potentially not very specific on a particular structure, but as soon as a galactose motif is present, then we will try to get rid of this protein. But I think also that maybe some processes are more specific, and I would look here more towards the antibodies, because of course the pharmaceutical industry also has an interest in figuring out which specific glycan motif is important for antibody function and receptor recognition. And I think there we know a little bit more which specific glycan motifs really trigger the immune cascade and which do not trigger the immune cascade. But yeah, that's definitely something I want to figure out, right? So
yeah, exactly. Yeah. I think it's like there's so much unknown, and then until we have the tools to look at it, we really don't know, right? So
yeah.
Thank you. That's great.
Great, thanks. We have a question from Patricia. She said, for those people trying to recreate the CIU experiment and do not have this specific instrument, do you think that it is possible to induce an external stress, e.g. heat, before the LC-MS analysis?
Definitely. I think there are a lot of ways to stress your protein. I mean, in the end, you always need an instrument that is capable of eye mobility to track these changes. But I think how you induce the stress is up to you. I think you can also try it offline with heat or sonication, or in what way you see fit, and then detect these changes. But then, of course, you need to be careful that you don't induce additional changes with your measurement. I think using the MS settings is the first way to try, because it's I would say the easiest way, because you can just ramp through your parameters without doing extensive sample prep. But I can also imagine that using heat, using sonication can induce stress, and will unfold the protein.
Great, thanks. And Etsky has a question here. It is interesting to study any of the found glycopeptide species groups which affect the disease. Do these play a role in protein function, for example, dimerization or further, and what exactly functional changes should we focus on in the next studies?
Not sure if I get the question. Can you maybe recap it briefly?
Did you want to unmute and just ask live your question? That may be easier.
Maybe I don't need to.
While we're trying to sort that out, I guess I had a question on the technology side. Is if a lot of your work you were looking at optimizing parameters, and some of these you came to kind of the limits of what the instrument could do.
Yeah.
What do you see as kind of the next couple years, what technology changes have to occur to make this more efficient and more easier for other people to implement this type of approach?
I think it's mostly the community that needs to change. Because I mean, we already have great instruments that are theoretically able of really nicely sequencing the glycan part and sequencing the peptide part. I think we need to make sure that the people are aware that glycopeptides are different, that glycopeptides are important, or that glycosylation is important. And yeah, mostly creating awareness that you need to consider certain aspects when moving from proteomics to glycoproteomics. I think in general, we already have good instruments. The problem that I was mentioning, for example, with the collision energies, is something that can be easily solved on the technological side to just be able to use higher collision energies. I don't think that this is a technical challenge, but more an awareness challenge.
Oh, it sounds good. So I think on that note that it's up to us basically. So
Yes.
I think we'll end there for today. Thank you again, Melissa, for enlightening us on the world of glycoproteomics. And I hope to see everyone next month. At the end of May, there'll be notice coming out. And as I mentioned on the short courses, deadline to sign up is a couple of days if you have interest in that. So thank you again, Melissa, and we wish everyone a nice day. Okay, bye-bye.
Bye-bye.